Hi everyone. Okay, so before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Just a second. So join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology. Okay. So just a second. Let me just check this. Okay. And the Telegram channel of Unacademy, ICSE and ISE both. So this link you will find in the description box. So go to the description box, click on the link and join this channel for updates regarding the classes. So we conduct special classes on Unacademy platform which are absolutely free. So learn from your favorite educator for free. Engage with educators via polls. Clear your doubt in the live special class. So basically we'll be conducting a quiz. So at the end of this session we'll be sharing the leaderboard so that you can know your rank and compete with your friends. So this one, that is clear your doubt in the live special class. So there is a separate question tab where you can type in your question, attach an image and send it to the educator during a live session. So whatever doubts you have, you can get it cleared during a live session as well. Okay. So for the top special classes, you can visit the link in the description. Now, Unacademy subscription feature, you can learn live from the comfort of your home. Unlimited access to all the courses, like different educators will come up with different courses, so you will have access to all the courses once you take the subscription. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. A regular doubt clearing session will be done, like after about 3 sessions, the 4th session will be your doubt clearing session. So whatever doubts you have related to the topics that are previously covered in these 3 sessions, so you can clear your doubt in this session. Okay, and also it is an answer writing session as well. Like let's say we are done with all the doubts and still there is a time left, so we can conduct a quiz or maybe we can discuss some question answers, so that can be done. Okay, then complete syllabus will be covered, mentorship and guidance is given, study material is given, the practice test, live test series, so you will have all of this, and along with that there are batches. Okay, so there is a batch in English, there is a batch in English, so you will have access to all the batches once you take the subscription. So depending on your comfort, you can go for any of these batches. Then there is daily practice section, weekly mock test series, live quizzes, daily MCQ and subjective test series are also there. Alright. So Unacademy plus subscription pricing. So since this is the very beginning of your class 9, so you can go with the 12 months of subscription. Okay, so if you just want to try like how exactly this works, you can try with 6 months as well. Okay, so this is completely your choice. And you can see as this is the beginning, so usually I recommend 12 months of subscription, and this is for 12 months it is around 17,250, and with the code, so you need to apply a code here. So there is a gift box here, right? So there you can apply a code Vani YT where you will be getting 10% discount on the subscription. Okay, so you can try this. And today's topic is about the prokaryotic cell. Okay, so before I start, first let's see what exactly is a cell. Okay, so today's session anyway will be discussing about prokaryotic. What is prokaryotic? Anybody? What is a prokaryotic cell? So why do we say that it is a prokaryotic? What is pro? What is karyot? Okay, so what is the meaning of this one? Just a second. No idea? Okay, So let's see this. When we mention, it is a primitive. Okay, so primitive type. One second. Okay, so is basically primitive. Okay, so this is a primitive. Karyote is basically a nucleus. Okay, so what is that primitive nucleus? That is old type of nucleus. Okay, so what is the meaning of old type here? The meaning is that basically, this particular genetic material or the DNA which is present, so this is not covered by a nuclear membrane. So there is no nuclear membrane covering. Hi Kashyap Astro. Okay, so there is no nuclear membrane covering. So that means you can find a DNA there. Okay, so let's draw this particular cell. So that is basically a prokaryotic cell. Okay, so in prokaryotic cell, so what all comes under the prokaryotic cell? So it is basically the bacteria, the mycoplasma. Okay, then other than that, you can also find, just a second, blue green algae. All right, so in this case, now let's say this is a bacteria. Okay, so here you can find a DNA. So there is a genetic material, but randomly this genetic material is present inside here. Okay, so there is no nuclear membrane covering this particular structure, and that's the reason we call these organisms as old type of or a prokaryotic organism. Reason being, there is no nuclear membrane. Okay, so freely this DNA is suspended in the cytoplasm itself, and wherever this DNA is suspended, so now this space is not called as a nucleus. So if a nuclear envelope is there, so that we call as a nuclear, nucleus. If an envelope is found, that is a nuclear membrane is found. Now here in this case, as there is no nuclear membrane, so we can call this space as a nucleoid. Okay, so this is a nucleoid where the genetic material is suspended. So this is completely in the cytoplasm. So this can be called as a nucleoid now. Alright, so let's have a look at what exactly is the cell, and we'll see certain features of a prokaryotic cell mainly. Okay, so this is the basic structural and functional unit of a life. So why do we say that this is a structural or a functional unit? Anybody? So why do we say that this particular cell, when we mention, so how is it a structural or a functional unit? So structural we say because it is forming a particular shape is given to an organism. Okay, so that is because of the arrangement of these cells. So there are many cells arranged in a different format. Okay, and they are forming a tissue. So many tissues together are forming a particular organ. Organ together is forming an organ system, and organ systems, many of the organ systems together will form an organism. Okay, so for any organism to be there, so main unit of structure is what? It is the cell, and hence we call this as a structural unit. Then what about a functional unit? Why do we say that this is a functional unit? Because these are the cells which perform different functions. So now if we are talking about epithelial tissue, okay, so what does the epithelial tissue does? It does the function of protection, it does the function of secretion, okay, and also it does the function of absorption. Okay, so many such functions are done. So that way, so epithelial tissue, the connective tissue, is distributing or transporting things. The muscle tissue, that is muscular tissue, is there for movement. The neurons, when we mention, so you can see the picture here. So this is of a neuron, a human nerve cell. So this will give you a sense of sense of perception, or we can say sensation. Okay, so that is mainly by a neuron. So in all these cases, it is a functional unit too. So now we are talking at the organism level, like organ system is there, organism is there. But if we are talking about a prokaryotic cell, so there is no tissue formation. Okay, so in case of prokaryotes, there is no tissue formation. Okay, and also there is no organ formation as such. There is no organ system formation. On the whole, the single celled organism, that is bacteria itself, is an organism now. So this is a living organism. So that means a single cell alone can do all the functions. So the single cell alone can perform respiration. Okay, so this alone can perform reproduction, this alone can perform digestion, this alone can perform circulation. So everything happens here itself in this living one or a living single cell itself. Okay, so there is no complex structure that is required. Hi Ridhima, good evening. So there is no complex structure required, but a single cell itself can perform all this function. And that is why we say that whenever a single cell is there, so it is capable of independent existence. Okay, so why do we say that it is independent existence? So that is because even though it is a single cell, so this prokaryotic cell can perform anything, like everything, and hence we call these prokaryotic cells single. So it need not be prokaryotic always. Even you can find a eukaryotic cell which is single. Which is that example can you give? Eukaryotic unicellular organism. So any examples can you give? Can you mention? So anyway, this one is a bacteria which I gave an example only for a prokaryote. Now there is a single cell, but it is a eukaryotic one which is of independent existence. So what is that? Anybody? No idea? Eukaryotic one. So eukaryotic, which is single cell. So under that we have many different, like amoeba is there. All right. So then you can find a euglena, all right. So all these protists which are there. So paramecium, and other than that the single cell, which is that single cell. So that is again with respect to eukaryotic, but most of the organisms are eukaryotic. They are multicellular, but the single cellular is yeast. Okay, so yeast is single cellular. So as such we have many different, just a second. Yeah, so as such we have many different organisms, and they are single cellular and they can perform all the functions just like the all other organisms perform. So now types of cell: prokaryotic, eukaryotic. So this is based on the structure of a cell. Okay, so let's have a look at the prokaryotic cell today. Okay, just a second. So unicellular organisms covered in a membrane. Okay, so there is a unicellular organism which is there, and this is covered in a membrane. So what is that membrane? So this membrane is basically a cell membrane. Okay, so outside cell membrane or a plasma membrane which we call. So outside this, what do you find? So there is a cell wall as well. Okay, so there is a cell wall. Okay, so cell membrane, cell wall. And again in case of prokaryotic, so there is one more covering, so that is a capsule. Okay, so you can also find a capsule. So now when we draw a prokaryotic cell, so let's draw this prokaryotic cell and see. Okay, just a second. Alright, so now prokaryotic cell, so there is basically a cell membrane. So let's see, this one is a cell membrane now. Okay, so covering the cell membrane on the outside, what do you find? So basically on the outside, so there is a cell wall. So let's draw a cell wall covering this structure. Okay, so now this part is a cell wall. So you can find a cell membrane, you can find a cell wall, and with that, so there is one more covering. So what is that one more covering? So it is basically a glycocalyx. Okay, so that is a capsule. So capsule, one more covering you can find. So 3 layers. So these are there in a bacteria. So you can see that how many layers of protection it is having anyway. Okay, so now embedded within all of this, you can find the flagella. So there is a flagella that you can see. Okay, so this part what you see now here, So let me just label this part, just a second. Okay, so now this part, what you see, so this is a flagella. Then what about this? So this is cell membrane. Okay, so this is cell membrane, this is cell wall, and this one is a capsule. Okay, so this is basically it is made of a glycocalyx, which is more of a carbohydrate itself. So now this is a bacterial cell. So I'll show you one of the cell which I have put here, just a second. So you can see this one. Okay, so what all is here? So there is a capsule, there is a cell wall, there is a cell membrane or a plasma membrane. So there is a layers of protection, and you can see two of the flagella that are drawn. So you can just this is gonna vary anyway. So there can be one flagella like this. I can just show you this. So flagella, let's say this is one bacteria now. So flagella can be only one, or let's say there is another bacteria. So flagella can be two on both the sides. Okay, so there is another bacteria which you can find a bunch of flagella at both the sides. Okay, or you can find a bacteria which is having a bunch of flagella only on one side. So this also is possible. Or there is another bacteria, so let me just show you here. So where you can find a flagella like throughout this bacterial body. So these are all basically what? These are all flagella. So this is gonna vary. So this is especially in a prokaryotic cell. So you can find many different arrangement with respect to a prokaryotic cell. So that is a flagella. Alright. So now you can see nucleoid. Why is it called as a nucleoid? Because wherever this DNA is present, so that space we call as a nucleoid. So now let's draw a DNA here. Okay, and with that DNA you will understand. So let's say now this is the genetic material. So let's just highlight that as well. Okay, alright, one second. Okay, so this part, this is a randomly this DNA is present here. Okay, so this is what now? This is the DNA or the genetic material which you can find. So now this part is actually the DNA or the genetic material. Okay, so wherever this DNA is present, and that space we call as what? So that space is called as a nucleoid. So this space is what? This space is nucleoid. Alright, and associated with this DNA, so you can find many structures in there. So you can find many of the ribosomes. So this is now a ribosome. Okay, so randomly they are present. It's not that it is present here at the corner itself. Okay, so you can find there are ribosomes. So now this structure is basically a ribosome. So you can see a ribosomes there. What is the role of ribosome? Anybody? What does the ribosome does? So you can see that with respect to prokaryotic cells, so range is basically, so what is the size? So that is with respect to 0.1 to 10 micrometer. So that is the diameter. Okay, so diameter can be up to 0.1 to 10 micrometer. So let's see, this is about 10 micrometer. So let's see here, this is the one which is of this is the maximum I'm writing. So this is maximum up to 10 micrometer diameter you can find. So bacteria, it can be 1 to 2 micrometer. So here this can be anything with respect to mycoplasma, or we can say this is gonna vary. PPLO, so this is this is a pleuropneumonia like organism. Mycoplasma is the smallest one, so that is without the cell wall. So let's have a look at the shape of the bacterium. So there is a coccus bacteria. What is that coccus? Any of you? Coccus is spherical like this. So this is spherical. This is spherical. Okay, so what is that? Bacillus. So bacillus is rod like. So you can see a rod like bacteria. So this is rod like. Okay, so rod like bacteria also you can find. So this one is a bacillus. So bacillus we will mention when, okay, so let's see this. So this is a bacillus, so this is for a single one. Okay, so if many bacillus are there, so what do we call it? So it is called as a bacilli. Okay, and here also if many are there, so we call it as a cocci. All right, so now you can see here, so if they are in a pair as such, so let me just draw it here and explain. Okay, so let's see, this is with respect to coccus now. Okay, so now two bacteria are there, so this is what, so this is, you know, two of the cocci shaped bacteria are there. So let's say some are forming a chain like this. Okay, so there is a chain as such, so this we call as a strepto. If it is forming a cluster, like, you know, random clusters are being formed, so this is now a cluster. So what do we call this? So this is a bacteria when two in numbers, when they are arranged two in numbers, so this we can call as a diplo. Okay, so what is this diplo? Diplococci. Cocci because they are spherical. Okay, so cocci is for spherical. Now you can see they are arranging in a chain, so this is for two in number, so when they are in chain, so we call it as a strepto. Okay, so this one is a streptococci, and you can see a cluster here. So whenever there is a cluster formation, so this we can call as a staphylo. Okay, so there is a staphylococci. So this is how you can see a arrangement. So strepto for a chain. Okay, and you can see tetrad, the tetrad cocci, the sarcina cocci, the staphylococci is irregular, basically forming a cluster. Okay, so same way this is with respect to cocci bacillus. Okay, or a cocobacillus is given as an example, and you can see different examples here with respect to Vibrio. So this one I just drew only with respect to a cocci. So if the bacillus also, if it is the same thing, so let's see this. Now, you know, let's draw one more here. So this part, okay, just a second. So now this is with respect to bacilli, which is a rod shaped one. So if they are for, like, you know, arranged two in number, so this is a diplobacilli. So if they are forming a chain like this, like, you know, there is a chain formation, so then in that case we call it as a streptobacilli. So if it is forming a random cluster as such, okay, so there is no like, you know, number is not fixed in this case, so this is forming a cluster anyway. Okay, so this is a cluster, so this we can call as what? So this can be called this can be called as a staphylobacilli. So these are separate now. So this is a diplobacilli. Okay, and the other one is streptobacilli. So this you will be studying in detail. So streptobacilli, and this one is a staphylobacilli. So that is based on the arrangement. So I'll show you one slide now, so you can have a look at the slide. Okay, so this is hi, good evening. So this is bacterial shapes are mentioned here. Okay, so you can see rod shaped bacteria is called as a bacillus. If many are there, so it is a bacilli. So now chain of bacilli is being formed. So what is that chain of bacilli called? So it is a strepto. Whenever a chain is present, so that you can call as a strepto. So this is a streptobacilli. So same way for spherical ones, this is called as a coccus. Since two of them are there, so diplococci, and they are forming a chain here. So this is a streptococci. Okay, so whenever chain is there, so you will be adding the term strepto. So streptococci, and here you can see a cluster. Okay, so that is a random arrangement, like, you know, randomly they are forming a cluster as such. So whenever a cluster is there, so that we can call as a staphylo. Okay, so this one is a staphylococci. So same way here you can see spirillum, spirochete. Okay, so these are the different forms that you can find in a spirilla. So this is completely gonna vary. Vibrio is comma shaped. Yes, so it is streptobacilli. All right, so now here in this case you can see Dakshak has answered. No, okay, so Vibrio, when we mention, so this is comma shaped bacteria. Okay, so any example can you give for a comma shaped bacteria? Any examples? Do you guys know which bacteria is comma shaped and what disease it causes in a human body? No idea. So this is basically Vibrio cholerae. Okay, the name itself says it is a Vibrio cholerae that causes a cholera. Okay, so it causes a cholera in humans, and this is basically comma shaped. So they are forming, like they are forming a bunch as such. They are arranged, and they'll be causing the disease. So this is the Vibrio cholerae. All right, so let's go to the next one. Organization of a prokaryotic cell. So what all is there in a prokaryotic cell? So you can find a cell membrane, which I already showed you. Okay, so there was the previous slide. I can draw it again, so I'll just draw it again and explain. So there is a cell membrane, so you can find a cell wall as well. So poorly defined nucleus, so that is, there is no well defined nucleus. Then you can see a plasmid in there. Okay, so what is that plasmid? It is a circular DNA. Okay, then other than that, the ribosomes can be found, mesosomes can be found. So let's draw here. Fast will draw. Okay, so far, like 1 second, I'll just take this color. Okay, so now this part is what? This is actually the cell membrane. So we'll draw the cell wall now. So there is attached to this, like in continuation. So we can say that there is a cell wall as well. Okay, so this one is a cell wall. Then surrounding the cell wall, you can find there is a capsule. Okay, so there can be the capsule here. Okay, so all these things are there. Okay, so all these things are anyway there, and with that you can also find there is this part, so that is the flagella can be found. Okay, so this is there. So now what is mentioned here? Cell membrane, cell wall. So one more thing is actually capsule that I have drawn here. So let me just label each one of them. So now this one is cell membrane, this one is cell wall, this one is actually capsule, which is made of made of starchy compound itself, or not starchy, carbohydrate like glycocalyx. Okay, so in that case, so 3 layers of protection is there, and then you can see what is mentioned in here: poorly defined nucleus. So why do we say poorly defined nucleus? Because this DNA which is present here inside, so that is randomly suspended without any covering. Okay, so you can see that the DNA is randomly suspended here without any covering into this one, like there is no nuclear envelope, and this space where the genetic material is present. Okay, so this is the space, and this space can be called as a nucleoid. Okay, so this one is what? This can be called as a nucleoid. So let me just erase this. I'll just take a pen. Okay, so now this space which is there, so space where the DNA is present, so this we call as the nucleoid, and this is the DNA. Okay, and now what else is mentioned here? So plasmid. So what is that plasmid? So plasmid, when we are drawing, so maybe we can just show a circular DNA over here randomly. So there might be a circular DNA that might be present. So other than plasmid, what else do you find? So there is a ribosome. So throughout this cell, throughout the prokaryotic cell, you can find randomly, the ribosomes are present, and these ribosomes have a role in protein synthesis. Okay, so this is there, and other than that you can find infoldings from this membrane. So now you can see this is the membrane, so from here you can find a folding. So this is called as a mesosome formation. Okay, so mesosome is found. So all these are the different structures that you can find. So what exactly does the mesosome does? So this is mesosome. So let me just label it first. So this one is mesosome, this is ribosome. Okay, and so ribosome is there. So this is a plasmid. So plasmid is a double stranded or we can say that this is circular DNA, double-stranded circular DNA. So you can find a plasmid in there. So all these structures, these are all organelles. So organelles are there, so along with all of this, and this part is basically a flagella. So this is a flagella now, which is helpful for the movement. So this is cell membrane, this is cell wall, and you can find a capsule. So let's see what we have missed. Yeah, so cytoplasm. So here, you know, this complete part which is there. So this whole cell inside this, you can find a cytoplasm in there. Okay, so there is a cytoplasm. So suspended in that a fluid, semi-fluid like structure, like, you know, in there itself you can find many of the organelles. So pilus, what does the pilus does? Any idea what is this doing? Pilus is basically, transfer of genetic material can be done. So if it is fimbriae, it is to hold on to the surface. So these fimbriae can be seen. So ribosome, flagella, plasma membrane, plasmid is shown here. So this is the DNA. So you can see plasmid is a circular DNA that you can find, and this is the random DNA which is present here. So many ribosomes you will see here. Okay, so here I have drawn only few, so you can draw more also, because ribosome does the function of protein synthesis. So this structure is completely like more of a ribosomes itself. So you can also show more ribosomes, so it's better if you show less. Okay, need not clumsy this particular diagram. Alright, so in that case, nucleus is there, which is a irregularly shaped region. So this is known as a nucleoid. So what is the nucleoid here? So irregularly shaped, one second. Just a second. Yeah, so irregularly shaped region, so that we call as a nucleoid. So that means randomly this genetic material is present there. Okay, so there is no nuclear membrane, so no nuclear envelope. So what is that nucleus here? So whenever we draw a nucleus, so we always draw outer membrane there. Okay, so there is a outer membrane as such. Okay, and inner membrane. So there is the outer membrane. So let's say this one is the outer membrane, and you can also see there are gaps there. Okay, which is actually forming that particular hi Abhinav, good evening. So you can find the inner membrane. So inner membrane, let's draw the inner membrane now. So this is now forming the inner membrane. Okay, so this type of nuclear envelope you can see in a eukaryotic cell. Okay, so this is now a inner membrane. So outer membrane and inner membrane together, so we call this as a nuclear envelope. So this is a nuclear envelope. So nuclear envelope is not found in a prokaryotic cell. So now you know these are fused as such. Okay, so these are fused as such, and you see there are gaps here. Okay, so what is this gap now? So this is only the one which is called as a nuclear pore. So this is a nuclear pore, and inside this what will be there? So there is a nucleoplasm. So let me just highlight that part. Just a second. So this whole thing which is present here. Okay, so inside here you can find a nucleoplasm. So suspended in the nucleoplasm you usually find the DNA. Okay, so this nucleoplasm now, so this is in continuation with the cytoplasm, so that is through these pores. Okay, so because surrounding, let's say here is the cell. Just a second, I'll just draw the cell. Okay, so let's say here is this cell. Okay, so now you know this particular nucleoplasm is in continuation, so that is with the cytoplasm. So here you can find a cytoplasm. Okay, so this is all cytoplasm. Now inside this you can find there is a nucleolus. Okay, and the genetic material is present where? So this part is the one which is having a genetic material. Okay, so that genetic material is nothing but the DNA, and that's the reason, so we call these, like, you know, this is a DNA, and that's the reason we call this as a eukaryotic cell. So why is it a eukaryotic cell? Because it is having a well defined nucleus. Okay, so it is having a well defined nucleus, and that's the reason we call this as a eukaryotic cell. So this is what, this is a eukaryotic cell. So here what do we mention? So there is no nuclear membrane, so that is where, so that is here, so there is no genetic material is not covered by a nuclear membrane. Okay, so naked genetic material can be found. So now what exactly is a plasmid then? So plasmid is extra chromosomal DNA. Okay, so this one is a extra chromosomal DNA. So that means you can find a bacteria. Okay, so randomly this DNA is suspended in there, and you can see there is this particular the circular DNA. So this is an extra chromosomal DNA which is small, which is circular. So you can see this is randomly present here. So this is big, whereas this one is small, and this is circular always. Okay, so here this can replicate independently. What is the meaning of that? So that means it can form one more copy of itself, like it can form a copy and it can transfer it to the next particular bacteria which is coming in contact. So that we call as a sexual reproduction in bacteria, and the method is basically a conjugation. Okay, so this you will study in detail, but anyway, the plasmid, what is this? So that plasmid is actually, it can replicate, it can form a copy of itself, and this can be transferred to the bacteria which might be there. Okay, so this provides a antibiotic resistance. Like what is, one second, just a second, let me just check this. Okay, so in this case you can see that the antibiotic resistance, so some of the, you might have read about if you take too much of antibiotics, so this is not good. So what's the reason they say that taking too much of antibiotic is not good? If you go on consuming, so that is also called as a drug abuse. Okay, so what is that drug abuse? Basically unnecessarily taking medicines like antibiotics especially. So what happens is these bacteria are having a plasmid. Okay, and they have this so they have this extra chromosomal DNA which we call as a plasmid. Okay, so now if you go on taking the medicines more and more, unnecessarily, so in that case the antibiotic resistant gene will be there. So that is in this plasmid. So this is the circular DNA. So there is a particular gene. So I'll just highlight that. So now only this portion. So let's say only this portion, what is this? So that is there for the antibiotic resistance. Okay, now as you continuously go on taking the antibiotics, so there is this gene which will develop a resistance, and even if a real disease, let's say really one is getting a disease and you are taking that antibiotic, but this is not working on that bacteria, this antibiotic is not working on that bacteria, that is because it has developed the resistance. It is it is having a capacity to fight that antibiotic. So this bacteria does not die, and hence usually when we mention over usage of any of the drug, especially antibiotics, is not suggested, because the bacteria will get adapted to that and they'll develop the mechanism where this antibiotic cannot cause anything in them, it cannot cause a death of, it cannot kill the bacteria, or maybe it cannot inhibit the growth of the bacteria, and that's the reason it is always avoided to go avoid overtaking of medicines. Kidney also be infected. So basically every organ can be infected by a bacteria. So in that case, so with respect to, okay, you are saying about antibiotic, yes, so that is basically why antibiotic is kidney is also getting affected, is it is too much of salt content, basically too much of the drug content is there, and this excess amount of drug that is there in the body, the kidney is the one which has to remove. So in that case kidney is also getting affected. Yes, that's right. Okay, so monitor bacterial transformation. So what is that? Monitor bacterial transformation. So in that case, okay, Thank you. So in that case, we can say that the bacterial transformation. So in that case, let me just explain you here. So this is a bacteria. Okay. So this is the surrounding environment. So this one is the environment. So now you know this particular bacteria can take up the DNA that is randomly suspended, or not just the DNA. So it is basically plasmid. So let's use the term plasmid here, not the DNA. Okay. So in that case, so here randomly the plasmid is present. Okay. So this plasmid can be taken into the body of a bacteria. So the bacteria can take up that plasmid from the environment. So let's say maybe some other bacteria might have been there and it is damaged. So this plasmid is freely suspended in the environment, and this can be taken, and that we call as a transformation. So you can see here monitor bacterial transformation. So transformation can also occur. Okay. So let's have a look at this. So cell envelope, the outermost covering of a prokaryotic cell. Okay. So this is a prokaryotic cell. So 3 layers are there in that. So one is basically, yes, my class is at 6:30. Yes. What's the time now? Okay. So this is still time is there. Yeah. So I'll anyway take the session, but I don't know. There was some technical glitch that was happening. So I need to check if this is if it is resolved, then I will be taking the class. Yeah. Okay, so now so 3 layers are there. So there is a glycocalyx layer, which we call as a capsule. Okay, so there is a capsule covering. There is a cell wall. Okay, so cell wall is made of what in case of bacteria? So it is made up of a peptidoglycan. Okay, so this you will be studying in detail, but it is not a cellulosic cell wall. It is actually a peptidoglycan cell wall. And then you can see a cell membrane. So as usual, all the cell membranes lipoproteins. So lipids will be there and proteins will be there. So all these things are found in a cell envelope. So you can see fimbriae. So what is the fimbriae for? Any idea? What does the fimbriae does? A bacteria is there. Okay. So this has to hold on to the surface. Like let's say it is here on the table, maybe here it is on the glass, or maybe it is on the rock. Okay, so to hold on to that surface, so this fimbriae is gonna help it. Okay, so hold on to that surface. So fimbriae is providing attachment to hold on to the surface. So fimbriae is basically holding structure. Okay, then you can see a chromosomal DNA which is randomly present here, and this randomly the chromosomal DNA which is present, so that space can be called as a nucleoid. Okay. Yes, so lipids and fats, it is the same thing. Okay, so it is the same thing. And the glycogen, pepsin and glycogen. What did I ask? So about the peptidoglycan. So this is, yeah, so fat and lipid, it is one and the same. Okay, so here in this case, so you can see this space where the random DNA is present. So that space is nucleoid. Then you can see a ribosome. What is the function of ribosome? Then you can see there is a plasmid DNA. So plasmid DNA has a role in sexual reproduction in bacteria, but it is not forming a multiple copies. So there are no copies or daughter cells formed here in the sexual reproduction that occurs in a bacteria. Only thing is that basically transfer of genetic material is done. So only genetic material exchange can occur, but there are no progeny production, and that is why sexual reproduction in bacteria is also called as a false reproduction. There is no progeny formation, only genetic material exchange that can be seen. So there are 3 methods which you will be studying in detail. So that is mainly about the plasmid. One is conjugation, there is transformation, and then there is transduction. So you will be studying about that in detail. The capsule is there, cell wall is there, and flagella. So flagella is for movement. So ribosomes to form the protein, small granule-like structures, so protein factory of the cell. Yes, that's right. Okay. So that means ribosomes help in the formation of a protein, and that protein is the one which is forming a particular feature of this bacteria. Now cell membrane is made of protein. So to form a protein, now this has to undergo binary fission. Like this is the bacteria now. Okay. So this bacteria has to undergo binary fission. So in that case, there is a furrow formation that is required. So if the furrow formation has to occur, like the cell has to elongate, a lot of protein requirement is there and a lot of lipid requirement is there. So protein synthesis, lipid synthesis will occur, and that is how the membrane formation can be seen. So membrane is being formed, so that is because that is cell membrane, okay, or a plasma membrane. So protein and lipid is required in there. So function of all this: protection from the injuries and the mechanical shock. So this is not with respect to only the cell membrane. So cell membrane, cell wall along with the capsule. So all of this together are providing a protection. So it is not budding. So in case of bacteria, it is binary fission. Like first the bacteria is there. So this bacteria is, let's say this is the size of the bacteria. Okay. So when it has to undergo binary fission, so this will elongate. So this will elongate itself, like it will become a little elongated, not larger as such. Here it anyway became larger. So it will get elongated. Then it will start to form a furrow. So now that you can see a furrow formation in the bacteria. So this is the furrow. Okay. So this is not budding. Okay. So once the furrow formation, here anyway there was a DNA earlier. So that is a plasmid. So this will duplicate. So that is divided. So there is an equal division of cytoplasm can be seen. Equal division of the genetic material, that is DNA, can be seen. Then this furrow will grow from outside to inside, and then both of the daughter cells are separated. So separation is done, and that is when we get two of the daughter cells. So now this is one daughter cell and this is one daughter cell. So parental cell itself is divided, and that is why asexual reproduction, when we mention, so identical copies are formed. Okay. So this is always identical copies are formed having the same genetic material that is copied and the cytoplasmic content also. Last slide, this one. So I'll just erase this. Alright, so let's go with the next one. So cell wall, so this is present between, just a second. Yeah. So this is present between the capsule and the plasma membrane. So this is a rigid covering. Okay. Then it is composed of carbohydrates and protein, which is basically a peptidoglycan. Okay. So structural support to the cell is given by the cell wall, determines the shape of the cell. So that is a bacterial cell itself, prevents cell from bursting or collapsing, because this layer, that is a peptidoglycan layer, what is it? So that is much more stronger. Now in case of a plasma membrane, so this is the innermost layer of the cell envelope and is semi-permeable. So here it was not written. So this is freely permeable. Okay. So cell wall, when we mention, it is always freely permeable. Like, it allows water also to enter. It allows everything, like, solute also to enter. So solvent and a solute can easily enter here. But in case of a plasma membrane, only the solvent can be allowed. The solutes are blocked. Okay. So if a solute has to be spent, so in that case energy is required. So in that case only the water is allowed inside. So let's say there might be calcium, there might be magnesium, so anything that is there in the water. So in order to take it inside this membrane, that is plasma membrane, energy has to be spent. ATP has to be spent, and only then this particular ions can enter. So helps in the interaction of a cell with the outer surrounding. Okay, so that is because there is a protein which is attached onto the surface. So if anything that has to be taken, so in that case there is a protein movement that can be seen. So helps in the interaction of a cell with outer surrounding. So in that case, one example I'll give you. So now, let's say this is a cell membrane, okay, or a cell wall itself. So in that case, anyway, you can see there is a protein that is attached, all right, and you can see this ion. Maybe let's take a ion itself. So ion has to be taken inside. So now, this is gonna get reversed. So how exactly this protein which is there, so this can move, okay. So this is the next phase, and you can see that whatever ion has to be there, so this is released inside. So that means now, as this protein was like this, so this has moved this way, so that the entry can be taken. So like this particular ion or maybe a particular thing that is required, or a protein, so that can be allowed inside. So that is by moving a protein. So interaction can be done. So composed of lipids and proteins. So this is mainly made of lipids, okay, and is made of proteins also. Lipids is nothing but a fat, okay. So proteins are also there, and these are the main components, but some carbohydrates also you can find. So along with that, some carbohydrates are attached as well. So this allows entry and exit of specific material. So this we know, so that is anyway it is semi-permeable. Alright, so let's see this. Which structure perform the function of, just a second. Yeah. So which structures perform the function of mitochondria in a bacteria? So here I did not mention that. So one more here you could see this. One second. This part. Okay. So here a mesosome is mentioned. Okay. So mesosome is a homologous structure, or we can say it is equivalent. Okay. So this one anyway, the question you need not worry. So mesosome, I think I mentioned, I don't remember. So this one, mesosome, is responsible for the powerhouse. Just like a mitochondria is present in our cell, that is eukaryotic cell, which we call as a powerhouse of a cell. So here also mesosome is just like that in case of a bacteria, that is a prokaryotic cell. So in that case, mesosome does the function of cellular respiration, okay. So why exactly respiration has to occur? So for any organism to survive, there is a ATP production is a must, because for performing many different activities, energy is required. So like to move a hand, or maybe to speak, then for the bacterial movement, so flagella it has to use, and then it has to move. So in that case, ATP is a must, so energy is required. So with respect to that, ATP production can be done by the mesosome, that is by a cellular respiration, which is equivalent to the mitochondria that we see in a human body, or basically any eukaryotic cell, okay. So here the question was, what was the question? Just a second. So which structure perform the function of mitochondria in bacteria? So you can see the option nucleoid, ribosome, cell wall, and a mesosome. So this is the correct answer. So mesosomes are just the inner membrane folding. So now cell membrane is there. So this cell membrane itself is folded like this, and again there is a structure. So now you can see many of these structures as such, okay. So now same way there is a folding again. So this is forming the cell membrane. So this is now a cell membrane. So these are the mesosomes, which are equivalent to the mitochondria that you can find in a eukaryotic cell, which have the function of cellular respiration, and after that the ATP synthesis can be seen. So that is energy production. Adenosine triphosphate can be synthesized. Yes, and the storage can also be done. Yes, so that is depending on the requirement that the energy is used up, rest of them is anyway stored. Alright, so protoplast or a protoplasm is without the nucleus, undergoing division, without the cell wall, and without the plasma membrane. So what do we call this? What exactly is a protoplasm? Any idea? So protoplasm, yes, so you can just mention the option if you know, or anyway I'll explain it, okay. So it absorbs energy and release in the form of a ATP. It is absorbing the energy. So it is basically oxidizing the glucose. Okay, so oxidation of glucose can be seen. Okay, and that oxidized glucose, what is it? So after oxidation of a glucose, energy will be released. Yes, so that is in a mesosome. Yes. Okay, so here protoplasm is basically without the cell wall. Okay, so living component of a cell we call as a protoplasm. So what is that living component? Now you know cell membrane is there, cytoplasm is present, and then you can find a nucleus. Okay, so this whole thing, so cytoplasm along with the nucleus covered within a cell membrane, so this we call as a protoplasm. Okay, so this is, now let's say this is a animal cell, so animal cell directly we can call it as a protoplasm. But for a plant cell, if a cell wall, like you know, in case of plant cell anyway cell wall is present, so we have to remove the cell wall, then only the remaining content, that is a living component, so that we can call as a protoplasm. So this needs to be removed because the cell wall is actually a dead component of a cell. Okay, so in that case this is not considered, but rest of the thing, so that is cell membrane, cytoplasm, and along with the nucleus, so all this together we call as a protoplasm. So protoplasm or a protoplast in a cell is without the cell wall, because cell wall is a dead one. All right, okay, so we are done with today's session. And before I end this, so let me just say about the batches. So we have a guardians batch, so this is for class 9. So you can watch the recordings as it has started on May 4th. So you have an option to watch the recording, but yes, you, like you know, many more sessions which are yet to go live, so you can attend the live sessions as well. And here the mode of delivery is English. Okay, so if you are subscribing for class 9, so you will be getting class 10 to 12 subscription absolutely free with us. And here also there is a Avengers batch which started on May 11th. So here again you have an option to watch the recording and many more sessions which are yet to go live. So you can attend the live sessions there. And in this case mode of delivery is English. Okay, so you can have access to these batch also once you take the subscription. So now in this case introducing community, so this is basically we have a community, basically never miss on any update from your favorite educator. So get session notes and practice material here, share your feedback directly with the educator via polls. Okay, then Bugs Bounty, so this is opportunity for all learners to report any inappropriate content in the video. Just a second. Okay, so you answered it as the, I saw now. Okay, so be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. Alright, so you can download the Unacademy Learner app, and this is my code VaniYT. Okay, so with this code you can get 10% discount on the subscription, and also you can use many of the free products that are there on Unacademy. So you can use the VaniYT code to unlock the free products on Unacademy platform. Okay, alright, so I'll end this session. So thank you all. So click on bell icon to get notified about the session. Subscribe to our channel, that is Unacademy, ICSE and ISC boards. Like and share the video and download the Unacademy Learner app. And if you need the PDF, you can join my Telegram channel, that is Unacademy. For my Telegram channel you can anyway join Vani Kari ICSE Class 9 and 10 Biology. Okay, alright, bye bye everyone, take care, thank you so much. Thank you.